The overall goal of this method is to tag proteins in Trypanosoma brucei in a high-throughput manner.
This method can help answer key questions in the kinetoplastid field about the localization and potential function of large cohorts of proteins. The main advantage of this technique is that large numbers of proteins can be rapidly and easily tagged in parallel.
Though this method can provide insight into localization, interactions of large cohorts of proteins can also be investigated by using other tags. For example, the BRA tag for a bioID proximity mapping experiment.
Demonstrating the procedure will be Ross Madden, a research assistant from our laboratory.
Prior to starting this procedure, pre-freeze a 96-well PCR cooler in the -80°C freezer. Prepare master mix A by combining the following in a 10 ml tube: 2,100 microliters of distilled deionized water, 105 microliters of PCR-grade DMSO, 105 microliters of 10 millimolar DNTPs, and 105 microliters of P-pot template. Pour master mix A into a reagent reservoir, and aliquot 23 microliters of master mix A into each well of a 96-well PCR plate. Using a P20 12-channel multi-channel pipette, add 2 microliters of 100 micromolar pooled primers to each loaded well. Seal the plate with film, place it onto the pre-chilled PCR cooler, and allow it to freeze for a minimum of 20 minutes at -80°C.
The freezing of PCR master mix A is critical to ensure an efficient hot start to the reaction, even when using a specific hot start.